Welcome, everyone, to Informing Seed Sourcing Using Fitness of Little Bluestem Populations in Minnesota. A couple of quick reminders before we get started. Please use the Q&A box on the right in the Whova screen for all of your questions. If you had to join directly through Zoom, you can also use the chat function in Zoom to bring your question over. The chat section in Whova is for any other comments that you might have. And because we know folks might have a bit of a hard time getting their questions in, Wes has been kind enough to say that he'll be coming back afterwards and answering questions that are on Whova. So if you have a hard time getting your question in now, please know that if you're able to get it in later, we'll still work to get you an answer. So today, our presenter is Wes Breaker, a PhD student in the Conservation Sciences program at the University of Minnesota in the Twin Cities. His work stems directly from an interest in natural resource management, and he puts his research directly into action through his work as the manager of the St. Olaf Natural Lands. With that being said, Wes, please go ahead and take it away.
Thanks so much, and welcome everybody. Today I'll be presenting a little bit of my PhD work, which is informing seed sourcing using populations of little bluestem in Minnesota. And with that, we'll get right in. I know we have a short session here. A lot of this introductory information, if you were tuned in to Jared Beck's talk earlier, and I know Dan's talk in the morning, a lot of it really touches on the loss of habitat, both worldwide, but very specifically regarding our prairie ecosystems. This is a huge issue when we're talking about preserving our native remnants, and it's a big reason of why we're all here today. So this map might also look familiar to you if you were at Jared's slide this morning. In this DNR produced map, this is a representation of prairie that existed in Minnesota prior to 1830. That's represented in the yellow, and that's the main component that you should be looking at in this map. Don't worry too much about all the other ecosystems that are labeled on there. The other contrasting thing that you should pay attention to in this map is specifically focusing on the red, which is bold, and that's our remaining habitat. And as Jared was talking about this morning, there's really less than 1% of this remnant prairie left in the state of Minnesota, and it's important that we're focusing on preserving it. If we zoom in on an example county, Nobles County, which is down in southwestern Minnesota, we get a really good idea of just how extreme of a situation it is. We can see there's only about 30 to 40 prairie remnants within this county. And when we're thinking about the topic of habitat fragmentation, or the phenomenon under which a specific habitat becomes separated from other like habitats by some spatial difference, this is a really big thing of concern when we're thinking about restoring and maintaining our natural habitats. So how do we combat this loss of habitat? Well, one way that federal, state, local, and private stakeholders have recently begun to think about more, and recent as in the last several decades, is restoring habitat on the large scale. And in Minnesota, at least, one of the most approachable and economic ways to do this is through putting seed that is collected back into a location. So collecting seed and planting it where we might be either converting over from an agricultural system or into areas that were already restored and need a little bit of help. Typically, plants are what we focus on. They're a critical part of the restoration, and they don't move around like a lot of other organisms we have. When we're talking about seed collection and restoring these plant communities to make habitats, we really need to be cognizant of genetic variation that exists not just in the biodiversity of these systems that we're restoring, but also within individual species. And this differentiation of populations within species can really differ greatly between geographically distinct populations. Additionally, when we're talking about restoration, it's absolutely essential that we take climate change into account when we're thinking about our seed sourcing practices. Our climate has already changed and it's continuing to change. Some studies and management practices have already begun to translocate species and populations specifically under the assumption that future predicted climates are what will make most sense for our future plant communities. And typically this has followed the predicted climate gradients that have been put out over the last couple decades. So with all of that said, how do we approach sourcing seeds to ensure that we have high population fitness and fragmented landscapes? In other words, how do we make sure that our restorations are sustaining and that they will be successful over the long decades to come? That's a really big question. And to answer that, we really need to look at a more in-depth question based on the idea that these populations that we are collecting seed from are very different from each other within their individual populations of an individual species. And so I'm moving forward with this question of, when we are sourcing these seeds from different locations, what is the scale of that variation across the landscape? To do this, I'm using a perennial graminoid species, little bluestem, Schizochirium scoparium. This is a common species across North America. Most of North America, it's wind pollinated. And from the restoration perspective, it's relatively ubiquitous in tall grass prairie restorations. It's also an important food source to both domestic and wild large herbivores, and it plays host to at least 11 different species of Lepidoptera. In addition, little bluestem is only second in terms of abundance next to big bluestem when we're looking at the variety and diversity of species that are in our tall grass prairies. Additionally, there has been little life history work carried out on this species along with other long-lived perennial species. If you tuned into Jared Beck's presentation this morning, he discussed at least 5 different species, and really throw little bluestem in there, that's getting pretty close to the number of species that have very, very in-depth life history work carried out on individuals, especially of that habitat, that lifespan, and that lifestyle. A lot of this work really has not been carried out because in order to do so, it's very time intensive and difficult to monitor individuals, especially in their natural settings. So as we're moving forward with this question of thinking about the scale of geographic variation, we really need to define what is geographic variation. In general, genetic variation within a species can be very varied across a population's range. If we look at Minnesota in this very homemade map of rough distribution of little bluestem in Minnesota, you can get the idea that a little bluestem population in northern Minnesota or southeastern Minnesota or central Minnesota, each of those populations has dramatically different environmental inputs put into them, and each of them has evolved under very different conditions. Just based on those assumptions alone, we could make a pretty good guess that each of them has a pretty big difference in their genetic variation. And specifically, when we're talking about genetic variation and how we're measuring that, we're talking specifically about the fitness of these populations. So why does plant population, or why does fitness matter in these scenarios? In the case of geographic variation in populations of little bluestem, we are using fitness as a measure of a population's ability to reproduce successfully. And this matters when we are interested in successful and sustainable habitat restoration. We want these populations and our restorations to be successful. And we can add to our restorations by being cognizant of geographic variation in our seed collection. So that's all great, but how do we go about measuring this geographic variation? It's important to consider that these plants come from all different conditions, and they are growing in different conditions that make it very challenging to measure specifically the fitness or that scale of geographic variation. Fitness is a proxy of current adaptation to a location, and it can be estimated using successive life history measurements of a representative population sample. So in the case of Little Bluestem, we can take a plant and we can revisit it every year. We can see, did this plant survive in this year? And if it did survive, did it flower? And how many flowers did it produce? And that's getting us right to that fitness definition of, is this plant reproducing? Which is what we are interested in here. And using a combination of these life history traits, we can calculate fitness that accurately describes the mean fitness of entire populations. It's difficult to attribute these differences in life history trait measurements to specific genetic differences in the wild, and so we utilize what's called a common garden. And a common garden is a plot design in which we have various populations represented, but in a common setting. We use this design to ensure that we're conducting this experiment in a setting that is as close to a situation where these populations might be experiencing in the wild. So for this experiment, we used 12 source populations from across the state of Minnesota. Each of these populations are from remnant prairies across the band of remaining prairie in Minnesota. From each of these populations, we took a representative sample from multiple families and we planted them into two separate common gardens. One of these common gardens is located in far northwestern Minnesota, and the other common garden is located in far southwestern Minnesota. So very, very spread apart in their locations. And what this is looking at is specifically each of these populations is moving a long distance from their source site. And we're putting them into these common gardens in two separate environments that each represent separate habitats. So you could imagine pulling a source population from southeastern Minnesota and planting it into northern Minnesota. We want to see what that sort of population looks like when it's put into such an extreme change in environment. Within this experiment, we conducted it over the course of 7 years, and within those 7 years, we tracked 1,356 individuals. Each of those 7 years, we revisited each of those individuals and took counts as to whether they had survived, and then whether they were flowering and how many flowers that were present on that plant, as I discussed in that previous slide. And then given these life history traits that we're measuring, survival, flowering, and how many flowers, we run models to compare how those fitness values compared to the distance that these populations were moved. And that gives us an idea about the geographic variation within those populations, within this Minnesota population of Little Bluestem. Additionally, we can extrapolate that out to climate change and pull in some environmental information. In this case, we look at precipitation difference. So that's the difference in precipitation that a population receives at its source site compared to the amount of precipitation that population receives in the common garden that it's planted in. This is a component of a larger project that my lab group is working on, and this larger project is the first time that this scale of common garden planting with translocating populations has been done with a long-lived perennial species. These methods in general are frequently used within quantitative genetic experiments on plants, and so the setup is quite common. The question specifically is what is novel here. And so when we take these populations and we move them, we've taken the life history traits and we've modeled out the mean population fitness that each of these populations experiences in those specific common gardens at both our southern common garden and our northern common garden. So if we take apart each of these graphs, on the x-axis, we have the distance in kilometers that a population was translocated and how far that source population is from the common garden. That common garden is located on that vertical line at 0. On our left is the calculation of the mean fitness of each of those populations. So when we compare these, we see there's a lot of scatter within individual source populations. But when we take the overall if we take the source population out of these models, what we see is a trend line that really represents what the geographic scale of the geographic variation is within these populations. So in general, what we get from this from these models is that we see that population mean fitness overall decreases as distance from source site increases. And we can think about that from the perspective of restoration. If you're sourcing for a restoration, it's very unlikely that you will be able to source seeds for that restoration from immediately adjacent locations. It's just not possible given the amount of habitat fragmentation, the amount of habitat loss that we've seen. And so what this information can do for us is help us think about what is an appropriate distance that we might source seed from in order to prepare this restoration for the future. And what this is showing is that really we should be looking at very, very local populations. It's hard to see in some of these source populations that are listed here. You can see little lines sticking out, and that's our standard error of each source population, which given how many individuals are within this project, it's a very, very small standard error when compared to the larger picture. So each of these points is very significant on its own. As a group, we start to see these trend lines forming despite this overall scatter of mean fitness. When we take this into the perspective of climate change, specifically using precipitation as a parameter to compare with, and knowing that we have these large changes already in our climate, we see a very similar story here. So in each of these graphs, we have our South Common Garden on the left, our North Common Garden on the right, and these are the same populations in each location. What we've done here is we are displaying our difference in precipitation between our common garden and each of the source populations on the x-axis, with that vertical line being the common garden. And we have our mean fitness again displayed on the y-axis. Each of the points represents or is the exact life history trait fitness calculation that goes into each of those, is giving us the mean fitness for each population compared to the difference in precipitation that each source population is experiencing. Just like with our geographic variation model, we see a similar trend of as you experience more dramatic differences, as a population experiences more dramatic differences from its home site, we see lower fitness overall. So population mean fitness decreases as the difference of precipitation from source site increases. An additional interesting point within these slides is if we look on this Southern Common Garden, there's a very large difference here between our southeastern sites in green compared to our Common Garden in the far, far south. The precipitation data that we used to compare with each of the source populations is based on 30-year averages of precipitation at each of the source sites. Our model line, the trend line in the middle, is representative of the precipitation during the 7 years of our study at the Common Garden. And so what this is showing us is there is a greater than 2 millimeter difference between similarly sourced sites and our Common Garden. Comparing this with our precipitation map that we saw earlier, showing climate change or precipitation changes due to climate change, that 25 millimeters, approximately an inch, directly correlates with the amount of precipitation difference that southern Minnesota has seen. And so that's just amazing to me that that is visible within the 7 years of this study. So to conclude, for Little Bluestem specifically, it would appear that local sourcing is best when we're thinking about the scale of geographic variation. Additionally, when we start thinking about distances and climate change and do we want to plant species from populations that more that are sourced from locations that represent our future climates, this evidence really does not support assisted migration in that case. Our southern populations, whether we're looking at geographic parameters or environmental parameters, our southern populations do not have higher fitness when compared with more local populations in the north. To conclude, this work is also supported by other research on 4 separate prairie forbs that my lab group is working on in the Healthy Prairies Project, and there's less specific trends that are visible, less clear messages. But what this research on forbs really suggests is that there's no directionality, but local sources are still performing higher than away sources. Local sources are still looking to have higher fitness than our away locations. So with that, I'll conclude, and I can take any questions.
Thank you so much for the presentation of your research. That was incredibly interesting. And assisted migration is something that I know our community has talked about a lot, the pros and cons. And so having some up-to-the-minute data that speaks directly to that is very exciting for us to be able to have at our conference this year. So one of the questions that a couple of folks had written in about was, so you were focusing on geography and precipitation as the variables that you were studying with your seed sourcing. Was there an indication that there might be any other extremely meaningful variables? They mentioned, temperature, timing of the freeze-thaw cycle. Obviously, you can't study everything at once, but could you speak to any of those other variables?
Yeah, I mean, that's an excellent point. There are so many parameters that exist out there that we could be measuring. As it so happens, I have a couple of extra slides I can throw at you all here. Really, the thing that we're looking at, so we test a lot of variables when we're looking at what makes sense and where we can see correlations going on here. In the case of geographic variation, we've looked at latitude and longitude and absolute distance are the main things we're looking at. But when we start thinking about environmental variation, we also see very, very similar trends within our temperature differences between our source sites and our common garden sites. The reasons we're using these 2 measurements specifically is, well, there's a lot of data out there that shows that these are very specifically correlated parameters when we're talking about climate change and when we're talking about site-specific variation. But the individual asking that question is absolutely right. There are so many variables that could go into this. Yes.
And we just had another question come in. So someone's wondering, how exactly do you define local sourcing? Is there a mile radius that you consider, or is that just a relative term?
Yeah, and that is the answer that was really put forward when this research was first thought of 10 plus years ago. Can we get a number on the distance that makes sense? And really, when we're looking at the geographic variation that is possible, there isn't a clear answer. I mean, we think about in your local area, if you were to do a restoration in your front yard and you want to source the most local seed, you might have fewer than 3 options for remnant populations in your area. And so it's just not possible to stick within a specific area. That said, when we look at the variation within source populations, such as in this graph, we see how much scatter there is. So we have our near populations, and that's going to be absolutely excellent. But we also see populations that have higher fitness as we move further away. They just happen to have higher fitness than our average populations. And so you're going to see genetic variation. The big thing to pay attention to, another take-home message here, is that if you're varying where your sources are coming from, that is going to play a really big part in the success of population fitness.
Mm-hmm. Thank you. So we had a different type of question come in, and this is even more speculative than the other ones I've asked you. But do you think you would have had different results if you hadn't used a widespread wind-pollinated species?
That's an excellent question. I would focus in on the wind-pollinated species aspect there, along with touching on another point. So Anna Peschel and Ruth Shaw, our PI in our lab, are looking at four different forb species, also prairie species that are they're pollinated by pollinators and not wind, and they're seeing similar results within our fitness and sourcing modeling. So the wind isn't playing a big part in that. Apparently, absolutely, wind pollination, and this is my speculation, probably results in farther spread of the genetics from certain populations. But additional research that I'm working on is also showing that even though little bluestem is wind pollinated, it actually is self-compatible and actually produces asexually. And so while you might have pollen from a couple hundred miles away moving in, you also have a lot of local, more nuanced things going on there. Yeah, I mean, we could do this research with pretty much any plant. What I'll say here and what Jared said earlier is there's only so much time we have, and these are really, really long-lived plants, and there's not that much research out there on them already.
Okay, so to change tacks for the type of question, we've had a couple of people ask specifically about the soil that you were using in your common gardens. Clay, silty, sandy, what was the mix there?
Yeah, so thinking about the common gardens and the soil. So each of our common gardens was done within a restoration matrix. So this is a agricultural landscape. We're not going to go put all of these non-local source materials into a remnant and do our research on them. And when we think about soil types, that's an extra environmental variable that we could be adding in here. We're not looking at that. There's too many variables. But part of what the Common Garden aims to do is to give a location, have a area where as many of those parameters are as even as possible for all those individuals represented within that garden. So while the soil type is certainly not the same as any of the remnant locations, it is relatively even across the average of that common garden.
Well, thank you so much. We have had more questions come in, but we're running low on time. And so hopefully you'll have the opportunity to answer those on the chat. But thank you so much for giving us this presentation today. It has been extremely interesting. And we're so excited to have all of our attendees at this conference. We're going to be having a break coming up, but please join us for future things. And I wanted to flag, you mentioned Jared Beck's presentation a couple of times. So for everyone who has listened to this talk and wants to check it out, that is also available through our platform. You'll be able to watch it for the next 3 months. Thank you so much. Have a wonderful day.